The world of biological research. Well, that's an amazing place to be in. But let me remind all of you before we get started. That's a tough place to be in. It's not an easy job to be in the lab. To do research, it takes guts. And more than guts, it takes skills. So get ready today. We are welcoming one of the most important person in our hands-on training who is going to help us propel into research. But before that, hello and welcome to Biotechnica's live stream. My name is Shekhar Sir. I'm the CEO and Managing Director and your success coach in the world of research. I hope all of you can hear me and see me clearly. Welcome to the session, guys. Welcome Shivam, Mandya, Koich, Dr. Prasanta, Abhirami, and everybody who is watching this live or recorded. We are right here to talk about research, right? So before we get started, let me tell you that today's session is going to be power-packed, super amazing, because I'm bringing the IISc scientist who is going to train you on hands-on. How amazing is that, right? Now, before I get started, everybody quickly in the chat box type down where are you from and what exactly you're doing right now so that I can customize this webinar, this live stream, according to you. How does it sound? Great, isn't it? So go ahead, guys. Welcome and hello, Sabrine, Rahul from LinkedIn. You're watching us on LinkedIn Live. Okay, people who are on YouTube, Instagram. Okay, wonderful. So welcome everybody. It is indeed a great pleasure to be hosting all of you today. And the topic of today is importance of hands-on training in biological research, right? Now, fortunately or unfortunately, our degrees are designed in such a way that, you all somehow get aligned so much towards research, so much excited towards research that when after your master's or bachelor's you don't get to do it, you feel disappointed. Isn't that right? Well, that's true. But sadly, at the same time, colleges' labs are not as equipped as it should be. And even if they are equipped, they don't have the right professors who could train you on high-level techniques for biology, right? So what if I could bring the best scientists in Institute of Science, IISc Bangalore, and train all of you? Well, that sounds like amazing, isn't it? So that's what we have done at Biotechnica, and today she'll be live with us very shortly. In the meanwhile, let me check where all of you are from. So now that we have the replies, okay, Andaman Nicobar, the first person, Shazida Banu, welcome. Priyanka from Ahmedabad, welcome. Then we have Murli from Hosur, oh, very near, Murli. Preeti working as a microbiologist. Where are you from, Preeti? Shrestha Barua from Bangalore, welcome. And Yashashwini, from graduate in biotechnology. Where are you from, Yashashwini? Let me know. Debangana from Kolkata, welcome Debangana. Vignesh from Chennai, Yazini from Chennai, Azim Ali from Jamia Hamdard. Okay, wonderful. That's a great place to be in. Ajay Savalia, Germany. Okay, we have people from Germany also joining. Welcome, Shivam. Welcome, Ajay. So Shivam is from Hyderabad, for a job in a vaccine manufacturing company. Wow, that's great. That's really exciting. Dr. Prasanta from Faridabad, looking for a job in R&D. Wonderful. Madhuri from Maharashtra. Murli. Okay. Abhishek from Purulia, West Bengal. Wow, that's a very important place in India's history, right? Anupam from Chandigarh. Sindhu from Bangalore. Okay, welcome Sindhu. If you get time, do come and meet us. Soham, Dombivali, Maharashtra. Okay. Ishita, Chandigarh, MSc Biotech, Gauri, Pune. Pallav Banerjee, R&D executive. Which company you work for, Pallav? Okay, then we have MSc Zoology, Sonam Jangra from Haryana. Snoopy, what's your real name, Snoopy? Because I see your comments and they're very Snoopy, so let me know your name. Somil from Ujjain. Princey Joshi from Ahmedabad. Wow, we have so many people. Soham, MSc Bioanalytical Sciences, PG, wonderful. Yashashwini from Bangalore, okay. Okay, the list is going on. I think I will end up reading your names if I just keep reading. Okay, Snoopy's name is Adiba Khatun from Lucknow. Okay, Snoopy, so I read your comments, Snoopy? Adiba, so Okay. Wow. Swati Arkare. Okay. Wonderful. So, guys, today's session is going to be all about you because my dream is to help you succeed. It's all about helping you succeed in your career, in your life. And truly speaking, this has been my passion throughout. And when this particular session was announced, I think 15 or 20 days back, so I was supposed to take it solo. I was supposed to just give my talk. Then I said, why not bring up Dr. Deepti Saini, who is an expert. But Dr. Deepti Saini ma'am is a very popular person and very busy person. It's very difficult to catch hold of her, and especially this season is for internships and hands-on training. So it's very difficult to get onto her. But finally, I'm here with her. So, ladies and gentlemen, boys and girls, let's welcome Dr. Deepti Saini ma'am. Welcome, ma'am. How are you?
Hello, everyone. Good evening. It's a pleasure to be here talking to all of you. If I were to choose a favorite place, my favorite place would be with students.
Oh wow, that's really encouraging and amazing.
Yes. When they say scientist, you would probably imagine a person who's like Einstein with lab coat gloves and on, and quietly doing their research. Of course, there are those phases where you're totally into your experiments, but
Right.
the pleasure that it gives me to interact with students and to transfer this enthusiasm that I have for biological research to students. That's
I,
my
mu- I
favorite.
must I must tell you that energy is infectious. Whenever energy is infectious, so that's really amazing the way you are. Like you are just amazing person. All right. So without much delay, our audience would love to know your academic and research journey so far. If you could just let them know through the ups and downs of your journey, quickly, ma'am.
You mean like my CV? Wow. So, having gotten my UG degree from Delhi University and master's from Jamia Millia Islamia, I got my PhD in biochemistry at AIIMS in New Delhi. Then
Okay.
spent a few years abroad doing postdoctoral research, and then came back to India, had a teaching stint, had my corporate job, working, taking care of a lab. Now we have a lab called, the name of our lab is Protein Design. And
Protein.
protein design is incubated by SID at IISc Bangalore. SID encourages students, alumni from IISc to set up their startups. Since there's a lot of research to be commercialized and for commercial use. So our founder director is Professor Rajan Dikhe. He retired from IISc after his 35 years there, and wanted to carry on his research for beneficial purposes. And with Synergies and me as a co-founder, we started up our lab, and it's an R&D lab. The whole idea of protein design is doing recombinant proteins. So we have three labs: the microbiology lab, the yeast lab, and the animal cell culture lab. And we do recombinant proteins in all of these systems, the prokaryotic system and the mammalian, and then in between the yeast. So when we think proteins, there are so many types of proteins that are there. Whether you think enzymes or hormones, antibodies, these are phenomenal proteins with so many applications in all the fields of biology. Right.
Mm-hmm.
So what we do is work on right from cloning a gene to overexpressing it to running downstream purification processes vis-a-vis chromatographic methods. Doing analysis of the protein in terms of its activity or things like that are all the kind of projects that we run in the lab. We also do PCR-based assays, gene expression analysis, and so on and so forth. So the fact is that proteins are our first love, but biology is DNA, RNA, and proteins. And now,
Right.
whether you are doing cell signaling studies, cancer biology, agricultural research, whatever, you're going to use,
Mhm.
you're going to see either at DNA level, RNA level, or protein level, or their
Right.
interactions. Right. So that makes molecular biology a very fundamental. It's not just molecular biologists doing molecular biology. Now we have cell biologists, cancer biologists, all sorts of scientists. You can't do without molecular biology techniques, right?
Right.
So
Right.
with that in mind, our molecular biology lab caters to all the three, like microbiology, yeast, and the membrane lab. Okay,
Mhm.
so now here's the thing. We do our own R&D. We also do contract research. But with the idea that in our days, we didn't get a whole lot of hands-on research till we actually got to maybe end of master's doing a small project, or in my first year PhD was where I did kind of independent experiments, which is handling all these techniques by myself. I know things have, universities and colleges are now very encouraging towards practical biology, and they
Mhm.
are also encouraging, industrial internships and students to get a whole lot of exposure to different techniques, what is new in the field, and everything.
Right.
Some of it is very cutting edge, but if you look at basic molecular biology, it's not rocket science. However, it's not trivial. So it's not like anybody can stand and start pipetting and experiments will work. Your accuracy, precision, reproducibility of results, all that depends a whole lot on practice. Okay,
Right.
doing looking at a demo practical during our degrees, or doing these group experiments where one person does the experiment, everybody else watches, is fine for degree, fine for knowing the principle of how things work. But then to me, it's pretty much like watching your mom make chapati.
Right.
Done. What happens when you
Right.
start doing it yourself?
Right. Of course.
Everybody
Right.
knows what happens when we start doing it ourselves, right?
Very
The,
true.
the shape, textures, the degree of cooking, everything is important. So any experiment that you approach is kind of like cooking. You know what the ingredients are, you know how much to put, you know what order to put them in, and you also know how much to cook them at every step.
Right.
It's exactly how molecular biology or any biological experiment works too, right?
Correct.
So till you actually get your hands on and start doing an experiment, the application won't come, or it won't be of any use just knowing a principle of how things work. So with
Right.
this thought in mind, we have opened up our lab to students who want to come and do hands-on. Okay. When I say that we have trained students in PCR, the expectation from the student side is they'll get to see how PCR works. From my side, the expectation is if you go to a research lab and they tell you you have to set up a PCR, you should know what all I'm going to require, how I'm going to approach this, how I'm going to set it up, and what kind of results do I expect. If things don't work, what is the troubleshooting I'm going to do?
Right.
Okay,
Right.
that's what is hands-on. So, and thanks to Shekhar at Biotechnica, we've been now fortunate enough to reach all of you in order to tell you all this, that you're most welcome to come to labs, attend the workshops that we are planning to run, and also have if you want to spend some time in the lab doing hands-on, internships for 15 days, a month, 2 months, you're most welcome. If you want to run your projects, UG or PG projects, that works too.
Right.
So
Right.
I think I'll let him talk a little bit about some of the plans that we have for these workshops.
Of course. Wonderful. So, students, now that you know she has amazing experience, more than I think 30 plus years of experience, even though she looks very young, 30 plus years of experience. Yeah, you look younger than me at times. So we have somebody really amazing within us here right now. So guys, go ahead, ask your questions, whatever you might have. I will display it on the screen and ma'am can answer. In the meanwhile, we'll proceed forward with our agenda for today, and that is importance of hands-on training in biotech and life sciences research. Now, to start with, whenever you are told to do a task in lab, it's completely different than how you do things, or the theoretical knowledge you might have read in the books. In the books, it's all in the 2D format, but when you really start performing in the three-dimensional format, there's a lot of planning which goes into designing the experiments, executing the experiments, and then analyzing the results and finding out where you went wrong. Because in the books, it just looks like, okay, this is how things are done. But it's very important to develop these practical skills. And it is also very important to learn these practical skills from experts like Dr. Deepti Saini ma'am. Now, just to give you an example, how to run the PCR in a lab independently, because that is what you're going to do. How to do a particular animal cell culture or ELISA technique. Now, many times you will feel like, okay, I know it, I have read it, I should be able to do it. But when you really stand in the lab, do you realize that reading about driving a bike and really driving the bike is two different things, right? Theory is not enough. You need practical hands-on, and you need able hands to guide you. And that is where her lab comes into picture. Now, here are a few things you should know. Ma'am has decided that she's going to conduct every weekend. So she's going to sacrifice her weekends for all of you. Okay. So ma'am has decided that she's going to conduct every weekend one hands-on training for each one of you. Now, I'm just going to share my screen and show across to you. I hope you can see it. So you have here, let's do it this way. The first one which is there is on 8th of July. It starts at 10 o'clock. It will end by evening 4 o'clock. And this is one day hands-on training on ELISA technique at Dr. Deepti Saini ma'am's lab. The next one, so this one is on 8th of July. Next is 15th July. She's conducting affinity chromatography hands-on training. And after that, 22nd July, again a Saturday at 10 a.m. It is practical RNA isolation training at IISc incubator lab at an online as well as offline. Fourth one, which we are doing, is on 29th of July. It starts at 10 o'clock and it will be again hands-on SD page training. And now all these, the benefit of all of this is something which you should know. The benefit of all of this is all these hands-on training are being conducted in Bangalore. But if you're not able to come to Bangalore, we will stream it live to you, just like how we are streaming it live right now to you. This exactly the same way we'll do the hands-on demonstration in the most detailed manner for the next 6 to 8 hours. So that, and ma'am will be herself explaining all of this. She'll start with the theory, then she'll go for the practical, and then she'll show you data analysis of all of this, right? And that is the benefit of this hands-on training. And being a very busy scientist, she has agreed, somehow we could make her agree that, okay, every Saturday, ma'am, please conduct these workshops. Now, let me tell you, next month, she may not conduct these workshops. Next month, maybe she is traveling to some, conference and she may not be able to do it. So please, guys, if you are interested, you can always register. The link I will request the team to share in the chat box. Okay. Now, quickly, in the meanwhile, let's look at some questions which we are getting. So, okay, so this is the Q&A time, ma'am. I will pass on I'll display the questions on the screen and you'll have to reply. So let me quickly pick out. Okay, so there is a question from Babita Devi. This is not exactly a research-based question, so it's kind of a career guidance question. I have to choose my graduation subject, but I'm confused between BSc Honors Biochemistry and BSc Prog Life Sciences in Delhi University. Also, BTech, Biotech, what to choose? I'm passionate about research. Ma'am, over to you.
Yes. As I said in the beginning, it is all very integrated now. It's not like biochemists don't do biotechnology or vice versa. Molecular biologists don't do cell biology. It doesn't work like that. So, the BSc programs are 3 years and BTech programs are 4 years. it also depends on your master's and further research, plans that you have to choose your program. You have to also go by which college you're affiliating with and what are the graduates from that college doing now. So the experience of your previous or of your seniors will also guide you to which college is best or which program is best. Spending 3 to 4 years doing a UG in biochemistry or biotech. See, biochem is a little more fundamental. Biotechnology is applications of molecular biology and biochemistry. So you would learn basic biology. The UG is, if you get a good overview of how basic biology works, then you can see which field interests you, what would you like to do your master's in. Say, for example, kids choose genetics, forensic sciences, they choose bioinformatics if you are more inclined towards programming or dry lab rather than do wet lab, like that. So any of these courses, depending upon how many years you want to spend, what college is offering the which courses reputed, please go ahead and make your choice.
Wonderful. That was great. So we'll go for the next question we have. Okay, this is already done. Let me just pull out some question. So I request all the students to start asking questions in the meanwhile. Next question. Okay. The next question is by Sindhu. Hello, ma'am. Before last weekend, I met you for qPCR training hands-on in protein design. Okay, great. So Sindhu can share her, experience.
Yes. Did you learn or did you enjoy the training?
Yeah, let us know in the comment section.
Yes.
Right.
Yes, we'd love to hear back from you.
Yeah. So now there is a repeated question which keeps coming. That is, will the certificates will be provided? Yes, certificates will be provided. The next question is, what is suitable if there comes a long gap between your lab experiments? What is suitable if there comes a long gap between? Okay, that's a question to you, ma'am.
So we're talking about a long gap. A long gap is very subjective. It could be a few days, a few weeks, a few years. So if for some personal reasons there are long gaps, I mean years. Some people, some students after their masters and all get married, also go on maternity leave and things like that. If that's the kind of gap you're talking about, don't stay out of touch with your field. If you read and keep up with online workshops and things like that of what is going on in the field, you'll still be informed. And when you want to get back to your experiments, it'll work, right? These are just pauses. These are not full stops, right? Except don't become redundant, because the field keeps moving further. More and more new techniques come up, new kind of research keeps going on. So you keep up with that. And you can make up for that gap that you've had. Now, short gaps are breaks. If you've been doing 5 years of intense research, say for example getting a PhD degree, you do deserve that 2 months of in the mountains where you can relax and recuperate from that. I don't know what kind of a gap you're talking about.
Right. So, students have shared the training registration link in the chat box. You can go and check it out yourself. Now, the next question is by Sindhuja. She's asking, what should I do if I want to join these training programs offline? So to answer that question, Sindhuja, all you have to do is the link which I have shared. You have to go there, choose the offline option. And then once you have enrolled yourself, you will get the address where you have to go for the offline training and the timings and everything, and we'll coordinate with you for the complete training process. Okay.
Absolutely.
Next question is from Ashika Jain. Should I choose B Pharma from DIPSAR or BSc Life Sciences from DU? Okay. It's becoming sort of a career counseling session. Over to you, ma'am.
Are you from DIPSAR? If it's close to home, pharma, BSc Life Sciences from DU is a good course. Then, I mean, in terms of courses, they are comparable. Then again, it's up to you whether you want to stay closer to home, you want to have an independent hostel life. A lot of UG students look forward to moving away from home and studying. As long as you get your UG degree and you are happy learning, The university is important, but then it's the important thing in UG is learning and enjoying that learning.
Right. So learning for that makes sense. Next question we have from Solanki Dhruvi, and she's joining us from LinkedIn. Her question is, I have completed my bachelor's in biotechnology, so I'm thinking to pursue my study in Canada in B Pharma. Will it work, or please suggest some master courses for biotechnology field? Over to you, ma'am.
Either works. Again, these are all personal experience. Everybody's situation is different. They are getting a master's degree again is enhancing what you have learned in your UG. So if you feel that you want to study pharmacy or in Canada, that works too. It all depends on what you want to do after that. Do you want to get a PhD degree, or you want to work in a pharma company? So those are also academia or corporate job is a choice that students have to make. Doing a master's in biotechnology in India is great. You take the combined engineering entrance test, choose a good university, or if you want to stay close to home, you choose a university which is close to home. These are some of the things that people consider while choosing their programs. If you really want to have a good exposure of how things work in the West, Canada is a great choice. You can stay back there for a PhD. You can come back here and do PhD. I mean, these are all steps and degrees. Again, what I'd emphasize is stay focused on the learning and enjoying the learning. Biology is important and hands-on.
Yeah, hands-on, right? Okay. So, next question, I believe, is something related to what you are doing. So Yashashwini is asking, what is the job opportunity that an MSc Life Science graduate can have? Can we work in protein production or in biopharma industry?
Definitely. I mean, now you know how things work. Everybody knows vaccines are important after this COVID thing and all. Diagnostic labs are important. Protein production, definitely. And there are specific companies doing enzymes only, or they're doing a whole lot of biosimilar molecules based on proteins and peptides. Biopharma industry, R&D, or if you want like a job, then again, biopharma industry is open for doing hands-on research.
Right. That answers your question.
As the courses are increasing, students are increasing, there is plenty companies and startups who are looking for skilled people to do research. So definitely there's work prospects.
Absolutely. It has definitely got a lot of scope. Next is Aarti Arul. She's joining us from LinkedIn. I'm working in an animal facility. We have to do health monitoring like list of pathogens, ma'am. Are you training for multiplex ELISA or multiplex PCR?
Sure, if you are interested, we could plan workshops based on that. Or if you want to come and spend a few days or weeks in the lab, we could definitely train you in those techniques.
All right. So I hope that answers your question. Definitely, we can do that. Next is, how is this course going to help for someone who is looking for a PhD? Well, it definitely will help, but over to you, ma'am.
If you feel confident that you can cast an SDS page gel and run it by yourself the first time you do it and do it perfectly, then go ahead and not take any courses. Okay. However, you will realize that if you have a prior exposure to a hands-on experience in a lab, it will help you get ahead in PhD. Otherwise, you will be spending time initially in your PhD learning techniques or learning hands-on to do independent experiments.
Right. So, to answer your question, let me make it very clear. Nobody is a born genius. We all learn here. And there's no harm in learning when there is an opportunity which exists, and that too from an expert like ma'am. The point here which I wanted to make is, whether you plan to do a PhD or whether you want to go for a job in industry after a master's, you will need these. If you have to stand in the lab, you should know how to drive the lab, right? Drive these equipments, right? So very simple.
Practice makes perfect.
Absolutely. Okay, Kishore Kumar Banerjee, how do you remember everything you read? Like, I forget everything every now and then. Ma'am, please answer this. Okay.
So no one's born with photographic memory. There are very few geniuses who can just read one topic and then remember point to point. I've been teaching almost 30 years and I still learn everything from every year. I learn something new from the students I meet.
Right. And to
Okay,
answer
there is always new things to learn. Plus, there is always books to brush up what you forgot. You don't have to remember all the details. You have to remember fundamental biology. Okay, the details Google Auntie knows all. You can always contact her and ask.
Right. I'll give you an example. I was in Vettur village and I had an opportunity to interview all these great people over there. Now, there were so many people that I forgot their names. So I had to refer to LinkedIn. Okay, what's his designation? What's his name? What's the company? So we all forget. We all have to go back. So it's fun to forget. Okay, there's nothing wrong in forgetting. It's just that we have to admit that yes, we forget, and then we have to just move on by learning more. Right? Okay. Wake Up has a question. The name itself is nice. Wake Up. So I'm a bit confused which master's degree is better if I had done my graduation in biotechnology. So you should give an MCQ Wake Up. But in the meanwhile, ma'am, over to you.
A master's in biotechnology would be the obvious choice, but that don't limit yourself. If you feel pulled towards agricultural sciences, please do that. Like I said, clinical diagnostics, forensics, bioinformatics, molecular biology, genetics. There are so many choices in master's courses nowadays. Life sciences, if you want to still make your choice, get your fundamentals polished and still decide on what your favorite part of biotechnology is. You can do a more a basic kind of masters also. Even till PhD, these are all training programs for you to learn the scientific method and become an independent researcher. They train you on how eventually you will set up your own lab and do your own R&D, whether in academia or as a startup, or of course you can always look after a lab in a big company as the R&D lab person, or eventually the head at the lab. So UG degree, PG degree, these are all stepping stairs for getting to doing further research. And training in doing research.
Very true. Very true. The next question we have is from Aisha Noor from LinkedIn, and she's asking, hello, ma'am, can you please guide how can we decide on which research projects we should be working during our last semester of master's degree? So again, if you want to do it at your college, see what all kind of research your teachers or your professors are involved in. Okay, if that interests you, you can do that. Otherwise, there in Bangalore itself, there are so many companies offering internships to students, or opportunities where they can spend some time in their labs and carry out their research. So if you have an idea on what you want to research in, you can contact these companies and see, I have this idea, can I run my research in this field? That, or else you can talk to those labs and see what kind of projects they do, and if that interests you, go ahead and do that. See, if you want to do something mammalian cell culture based, toxicity studies, or cell proliferation kind of studies, or you want to see antimicrobial screening you want to do, or you want to do some cloning and expression purification. These are some of the ideas that we give for students who want to come to our lab and do a master's dissertation. I've had about 15-20 students who have done their master's 8th sem, 4th sem project at our lab. Right.
So if you have an idea, run that. Otherwise, you see what kind of projects are offered, and pick one of those up.
Right. I would also like to take this opportunity to let you guys know that ma'am runs a lot of internships. So if you guys are interested, we are doing a molecular biology techniques internship, which is starting from 10th of this month. And it's a hands-on internship. Already seats are full, so she's taking only 10 students. So there are 3 batches as of now of 10 who are going to go in. First batch goes in July, second goes in August, and the third will probably go end of August or September. So we still have slots left in the September internships. If you are interested, let us know. We can always plug you in there. Okay.
Like a couple of weeks, 15 days, kind of basic molecular biology techniques.
Yes, we'll be training you on more than 15 basic molecular biology techniques over there.
Yes,
right?
yes, absolutely.
Yeah. And then we have now next question from Sachi. How can we update ourselves in the subject of our interest, especially the latest techniques and also during gap periods? Follow Biotechnica's, enroll in Biotechnica's courses, you will get it.
I was going to say that. If you want to keep updated, you have to join Biotechnica's WhatsApp group, keep up with their emails and all the links that they send. Okay. And this is also my favorite question, because nowadays everything is available online. Like students spend a lot of time on even social media platforms, right? Instead of just browsing through, funny or your hobby kind of reels and videos, please check out some of the stuff that has been put out on YouTube, Insta, where pertaining to biotechnology. And you can read up online on what's the latest in terms of the techniques and the kind of new developments that are coming up in the field. Even 15 minutes a day, if you spend looking at very beautiful videos, some of the biotech related pages have on them. Biotechnica's page too, you should follow and keep up with.
Right. So to answer your question, Sachi, just follow us. No one else, just follow us. You should be fine. Okay, we'll take it.
Yes.
Okay. Next question from Pallav. Ma'am, I'm an executive in R&D. My aim is to start an industry. Does it necessarily do PhD? My answer is no, but over to you, ma'am.
Another one of my favorite questions. See, you have a PhD startup founder, and then you have a non-PhD founder here. And it's not like it takes a degree to make a success. In fact, if you, like us, wait till you're close to retirement to start up, your energies are not as high as you might have in your 20s and 30s. I'm serious. I'm so impressed with people who are starting up now really early. And then getting the whole infrastructure set up, and then having experienced scientists come and actually help with the whole execution. The founder or the person who starts the company or the industry doesn't have to do everything. You have to build a team who will do what you have a vision for. So you don't have to start. You don't necessarily need a PhD. There are so many small government grants which are aimed towards bringing up students' ideas, Yuva and the Tare, and then there is there are these ignition grants and all, which are open to individuals who have an idea and want to run it. So yeah, for sure, a PhD is not mandatory.
Yeah, and there's no age to start a company. Probably
yes, and there is no age to start a company.
There's no age. You can start any year, any day. Even if you are 60, you can start. Even if you are 20, you can start.
Yes, but in the West, you've heard of these 14-year-old millionaires, right? They had an idea and they know how to commercialize it. Again, build the right team to do it for you.
Yeah, so that answers your question, Pallav. Next is from Kalyani from YouTube. Okay, please tell me the future of bioinformatics. Okay, I've told that many times, so you should tell. Yeah, go ahead.
Yes, bioinformatics is a wonderful area with all this, big data coming up, genomics, transcriptomics, proteomics, metabolomics, all these omics, and comparing them, making sense of how the whole, orchestra happens in a cell. Can be done using bioinformatic tools. The comparison between how things work at DNA level, RNA level, and protein level, and how the metabolites work, everything you can study using bioinformatics, big data analysis, clinical research. There's a whole lot of scope to doing bioinformatics.
Right. So that answers the question, Kalyani. Next question is Nofia. Okay, before we go to Nofia, I have a story to tell all of you. Okay, so this story is all about like how I met ma'am. Okay, so Like we have a Bangalore Biotech startups WhatsApp group. So she was a part of it. I joined new because I'm very new to biotech industry. So I was not aware that there exists a group. So I joined, and then, okay, we were just interacting, but we were not talking directly because I didn't knew. So they meet every month probably. So I was there in the meeting, and so there's this lady sitting, very dynamic, very bubbly, very enthusiastic, and so we started talking. Somehow we resonated our thoughts, and then I was like, okay, so can you train my students? And she was like, would you send them to my lab? I said, I would love to. And then we were like, okay, let's do this, right? And that's where it all started. And I must thank you on record in front of all our subscribers that it was a game changer. Thank you so much for allowing my students into your lab, because the things which you are training is really valuable, and your vast experience is coming very handy. Like the students who are getting trained under you, they're coming back to me and saying, wow, this is something which I wanted. In fact, the other day I had a student from Dubai, so she attended it in a different time zone, virtual hands-on live training, and she WhatsApped me that, sir, now I feel confident that if I stand in a lab, I can do this. That's an amazing feeling that a student who could not come here, she's in Dubai because her family is there, but she can now do her research independently, all thanks to Dipti ma'am. So students, if you have this opportunity, please grab it before ma'am gets busy with her projects. Let me remind you, she is a very busy person. She gets a lot of projects from industry, and she may not be available next month, next week, or whatever. So as long as you have the opportunity, grab it. Okay. So let's bring in the next question now. Naufia has a question. Ma'am, can you explain about the scope of future cancer biology? Okay. Over to you.
Yes. So since just to continue from where Shekhar was saying, I'm actually very happy to collaborate with Biotechnica and welcome students to our lab. They have been supporters and encouraging biotechnology students to get more informed and do the best in their careers, and they have been aiding students in all possible ways. So if our lab could contribute to this, one person can't do all the research possible, right? So what I couldn't do in all of my time spent in research, I'd like to hand it over to the next generation, like I said, to spread that enthusiasm to the next generation, so that they can take up become interested in biology and do good research. And thanks to these ARM and WISE fellowships that Biotechnica has announced, there are a lot of students who are showing keenness and spending time in the lab and learning hands-on, so that they can be ready for further research in their careers, right? So when it comes to cancer biology, see, there are things that we are still learning to understand. When 2003 human genome was announced, the idea was by 2020 we'll have everybody's genomes sequenced, understood. We'll have personalized and precision medicine for everybody, right? But 2020 came, with it came COVID, and we are still nowhere any close to that whole personalized medicine, except a whole lot of in-depth studies have been done as to how cancers probably arise, how they change forms, or how they metastasize, how they become more aggressive and lethal, rather than being just benign tumors or being a lump of cells which have gone rogue. So, but still, every type of cancer is different. The cancer you'd have as a lump in the breast, or is like totally different from lymphocyte based or a blood cell cancer, isn't it? So
Right.
we are still understanding how cancers arise, how they spread, or what can be done in order to control them. We have better diagnostics to early diagnose them, and we are moving towards therapies. Okay, chemotherapy was so random, so many side effects, so bad. But now with immunotherapy coming up, making it little more precise delivery of drugs to the cancers, things are going to get better. I think a few months back there was this study where 12 colon cancer patients were treated with an immunotherapeutic drug, and all 12 of them their cancers regressed. So
Right.
yeah, I mean, I think, person who's had a relative or a distant relative succumb to cancer, it's so prevalent, has this dream that one day I'll find a cure to biology. I have a whole lot of students who've come in with that I want to find a cure for cancers. It'll
Right.
be awesome. There is plenty scope of research going around all over the world to find a cure for cancer or to stop it from spreading at early stages itself.
Right. So for Nokia and all my audience, I have a point to make. Okay. As long as the cure for cancer is not found, your future is glorious. And the day the cure is found, your future is even super glorious because you will have experience that you can implement in the lab and create treatment for cancer. The whole world will come and bow in front of you if you have a treatment or if you can create a treatment from the SOPs which exist in the world. So it is worth investing your life and career into cancer research because that's the next big thing. And that is why we're conducting a workshop on cancer biology this month. I think it's on 22nd. It's another Saturday. And it clashes with Deepti Saini ma'am's training also. So you'll have to be mindful which one you choose. But yeah, we do have a cancer biology workshop coming up. Okay, so I think we have spent a lot of time on this question. Let's move on to the next one. You have to register for this training. The link is given over here. Okay. Now, the next is, okay, I think we have answered this question. Let me quickly jump on how to join online lab training. Do I get an email link? Yes, you'll get a link to join and you'll be able to see it live. There'll be all online live demonstration and ma'am and her team will be doing it for you. Okay. Next question. Recently, I've completed my master's in microbiology, Sapna. So now what should I do? What are the relevant scopes, please suggest? Master's in microbiology. Over to you, ma'am.
Again, this is an individual choice. I'll have to keep it, reiterating that if you are not, if you are looking for a job, there is plenty scope to find jobs in microbiology labs, and not just restricted to microbiology. You can always look for jobs in pharma labs and diagnostic labs. With that in mind, if you are interested or passionate towards biological research, in that case, I'd highly suggest that take the CSIR exam, get a fellowship, and go to a good university and get a PhD. Okay.
And take
Yes, absolutely. So depends on what your scene is. I mean, if you need a job, go ahead. You can get that experience. Of course, you can always come back to a PhD later. But here's my 2 pence worth. If you want to really get a high degree or continue your research, do it without breaks. First, get those degrees, then get job experience.
Wonderful. So next question is from Relieved Hat 4. What's your real name, Relieved Hat 4? I'm a biotech first year student, would like to know the thought process behind coming up with project ideas and research papers and the limitation that should be considered. Oh, technical. Over to you.
See, even science involves a whole lot of art. Okay, it has to be interest first. So if you are interested in a field, you read up research papers and do literature survey as to what all kind of studies have been done on that topic. Okay, I am 100% sure if you read 100 papers on the area that you are interested in, you will come up with questions, and you will be able to creatively come up with a plan of how to study that question.
Right. Now, let me commend Relieved Hat 4 for thinking of all this while he's in his first year BTech or bachelor's. That's a great place to think and do all this. So that's a great question to ask for. Next is Kalyani. I have a confusion between veterinary science and bioinformatics in bachelor's degree. Okay. Over to you. Okay. Veterinary science and bioinformatics, which one we should choose? Over to you, ma'am.
Again, it's your choice. If you are passionate towards animals and veterinary, diseases, or veterinary, animal husbandry, or whatever your interest lies, if you are passionate about that, please study veterinary sciences. And
Right.
that's more hands-on, wet lab, kind of thing. Again, bioinformatics is a lot in silico and computer-based. So those are
Right.
also some of the things people consider while choosing, lab work or field work versus a desk, kind of research. So bioinformatics will be a whole lot of computer-aided studies.
Very true.
So and then you're saying a degree. So I'm assuming masters. Yeah, again, it's again a very fundamental degree where you should learn and enjoy the learning.
Right. So now before we take the next question, I would like to, show up the agenda for which we wanted to discuss today. So the benefit of doing a hands-on is, of course, you gain the familiarity with the lab equipments, and that leads to better results, better experiment designing, and that obviously brings me to the next point, which is experimental designing and troubleshooting. Because many a times there can be some basic errors which you're doing, which leads to a bigger disaster in the making. So it's always very important that you are doing some experimental design planning and you know how to troubleshoot your experiment. And that is where, training is required. You require a hell lot of training to do your experiment successfully. Otherwise, you'll keep doing the same mistakes again and again. My suggestion is always learn from an expert and learn from their mistakes, and so that you become a better scientist, a better researcher in the future. Now, very important aspect is data collection and analysis. And I'm sure ma'am will agree with me. Even if you have performed your experiment in the better way, data collection is must and analysis is even more important. Ma'am, would you like to throw some points on this? Data collection and analysis after the experiment.
Yes. So wet lab doesn't mean that you don't look at your data and analyze as to what it tells you. So, for example, when you're doing a qRT-PCR experiment for doing gene expression analysis, once you run the PCRs, then
Right.
you have to look at what kind of outputs coming, right? What kind of values you get from here, because we are saying quantitative. So then we have to quantitate the fluorescences. And then if you're studying for gene expression studies, you have to compare and see. You're checking for upregulation or downregulation between samples, or even multiple samples you're comparing in. So there comes the analysis part, because otherwise just running an experiment will not make sense of what your biological question is. The most important is what is the question you're asking, and when you do an experiment, what kind of answers you expect from it, and does your data confirm your hypothesis or makes it null?
Yeah. So the next point which I wanted to make is laboratory safety and ethics. You look at this. There was an accident way back in, I think, 2007 in IISc Bangalore itself, I remember. And after that, there was a huge hue and cry that researchers get exposed to so many harmful chemicals. So it's very important that lab biosafety as well as safety is maintained, and it's mandatory. Now, you cannot read about a manual and say that, okay, I know lab safety, unless you learn how to practice it. And that is where hands-on training comes into picture. You really learn how to handle equipment. You really learn how to handle chemicals and reagents and how to use them, how not to harm yourself and your fellow co-workers while handling them. And same with the ethics part when you're handling lab animals, and a lot of experiments where bioethics is involved. You should learn all of this, so that when you go into your PhD or your research lab, you are fully independent and aware of the facts, and nobody comes to you and says that, hey, you did a mistake, or you did you broke a rule by not following these things. It should be there imbibed in your DNA. Lab safety and ethics is very important. What's your take on this, ma'am?
Yes, absolutely. I mean, experiments are important, but the researcher is more important. If the researcher is safe, they will do more experiments. And
Of course.
to learn, and to learn how to, so we work, unfortunately a part of the game is that you are exposed to mutagens, carcinogens, teratogens, so and so forth, pathogens, infectious diseases. Yeah, I mean, diagnostic labs, I've heard of accidents where despite the double gloves and everything, people have poked themselves with hepatitis injection sample or things like that. Yeah, I mean, hepatitis is one, HIV and all. I mean, that's why you see all those, the PP for protection for working in COVID labs, right? You don't want to accidentally get affected. So how to properly, work in the lab such that you are safe and can do more experiments is also a very big part of training. Good laboratory practices are initially taught to all students who spend time in the lab, so that they keep safe.
Right. So last 3 points I'll quickly make before we end this session. You also learn communication and collaboration. Now, many a times, unless science is communicated in an effective way, it's of no use. And that is where this point comes into picture. And of course, you cannot achieve science, you cannot achieve research without collaborating with other researchers. Like you can say, I'm collaborating with Deepti Saini ma'am, so you all are getting the training, the valuable training. Otherwise, she is very busy with her own company. She will never bother. But she has this passion to train the student, and I have this passion to train the student. So we have an overlap. So we are collaborating. The same way, when you're doing research, you'll have passionate researchers in other labs. You may have to communicate, collaborate, and reach out and get the job done. So this is very, very important for all of you. And you should know that collaboration is the key to your success in the research world. Okay. Next one quickly, because we have last 3 minutes left, real world applications. So, okay, you do your, my mentor once said that either you sweat in peace or you bleed in the battle. You choose your choice, right? So we have to decide whether we want to train ourselves now and then do our experiment successfully when we are really in the lab, or we just bleed in the battle. So real world applications of hands-on training is huge, and of course it helps you in career advancement. That's my last point for all of you. Now, we could take a lot of questions, I believe, in today's session. However, I'm sure that we missed out on some of the questions. So all you have to do is, this is my email ID. You can drop a mail to shekhar@biotechnica.org with all your research questions, which we could not cover in today's session. And if you're watching it recorded also, you can still email me, no problem. And if I know the answer, I'll reply it. If it needs to be forwarded to Dipti ma'am, I'll forward to her and get a reply and fast forward to you. But most important thing here to all of you, you have to know this, that no dream is too big. Everything is achievable. Everything is doable, but it's how we collaborate, it's how we coordinate, and it's how we decide that we are not going to give up in our dreams. We are going to pursue our dreams, we are going to execute our dreams, and we are going to win over our dreams. We are going to win over bigger dreams, actually. Ma'am started this lab with a dream to do a lot of protein design, protein engineering, and then she had this inbuilt inherent passion to train students. She loves teaching, and that is why now that she has opened her lab for all of you, my suggestion is take the advantage before this opportunity closes, because she takes only 10 students per batch, not more than that. Okay, so keep that in mind. And thank you so much, ma'am, for joining us today. It was a long 1 hour, I know.
It has been wonderful interacting with the students, and I'd really like to see them in the lab and doing good research in the future. All the best.
All the best, everybody. Take care. See you soon in the next one. Till then, bye-bye.